Well, hello everyone. My name's Julia Cooper, and I'm working on the live seeding project on Work Package 2. And this is one of our modules that we're using for training within Work Package 2, all about training on organic cultivar testing. And within Work Package 2, we're developing 5 different modules that will give you the skills to implement organic cultivar testing. So introduce you to lots of the trial design, data analysis, and tools involved. So this particular module is called Participatory Trial Methodology, and it's divided into 4 units. I'll be talking about those units in a minute. This is a module developed in collaboration with my colleague Charlotte Bickler at Organic Research Center, and also Frederick Ray and Ambrosio Costanzo, who are at ITAB. So just to cover or just refresh our minds really on what we're going to be doing in this particular module or module, yeah. So you can see this image, which is taken from one of the outputs of the Live Seed project, which was a predecessor to Live Seeding. And we'll talk quite a bit more about that during this module. And this shows the 4 key aspects of participatory on-farm cultivar evaluation trials for organic low input systems. And you'll see in the bottom right there, experimental design. So that's what we're really going to focus on in this particular module. And then module 9, which follows this one, is about data collection and management. The other 2 quadrants above we're not covering in the modules in this particular set that you're listening to right now. So let's learn a little bit about the module. Okay, so I'll just give you a little bit of an overview of the module. I just wanted to explain that this highlights work done in the Live Seed project, which was the predecessor to Live Seeding, the current project. And Live Seed was a Horizon 2020 project on participatory cultivar evaluation approaches that produced lots of really useful outputs. One of them is shown there, Ray et al. 2021, Frugal multi-actor and decentralized cultivar evaluation models, which was a deliverable from Live Seed. And if you have a copy of the PowerPoint slides for this presentation, you can use that hyperlinked link there to navigate your way to find that deliverable and download it. It's quite long and detailed and really good document that you might want to read over when you've got time. But there's also these 2 booklets that were produced in the same project that summarize quite a bit of the content of that deliverable. So this Buck et al. Guidelines for Organic On-Farm Cultivar Evaluation Trials and the Riviere et al. 2021 Selecting the Appropriate Methodology booklet. Those 2 also, there's a hyperlink you can find on the PowerPoint slides to navigate to those and download those. So this module is using those resources as well as the case studies from the Live Seeding project, which we'll talk about a bit later. So yes, please download those now. So this is the details on what the module is about. So the overall goal of the module, all about providing guidance on how to set up participatory cultivar evaluation trials. And we've just got this sort of mention about who the trainees are, the trial managers, giving them the skills to set up, assess, and manage a successful trial, and also know about advantages and disadvantages of different types of trial design. And at the end of the training, the trainee will be able to follow the steps required to design a participatory cultivar evaluation trial based on the principles of frugality, which we're going to talk about a little bit more in a minute. So our target learner is really these researchers or trial managers who have some basic data analysis skills and understanding on experimental design, but aren't, really advanced at that, but just know enough that they can sort of follow the content of the module and sort of know the right questions to ask when they need to bring in experts for further advice and guidance. And these are the module learning objectives, so a little bit dry to read, but just all about how we're going to learn about different cultivar evaluation methods with on-farm decentralized evaluation, principle of frugality, as I've mentioned, objectives, constraints, methods, and using these decision trees that we're going to see in this unit about selecting the right trial design, and a little more on frugal innovation. So those modules, those objectives actually sort of are woven through the different units within this module, so that you'll sort of get the skills, to have achieved those learning objectives through all the units. It's not a separate unit on each of those. So you can see here the structure. We're in this 8.1 right now, which is about moving from traditional plot trials to frugal trial design. And then we'll go on into tools and methods, and then the case studies and the practical tips. So through those 4 units, hopefully you will have achieved those learning objectives. How are we going to learn? So here we are now in Unit 8.1. This is just the outline, what we're going to cover in this particular unit, talking about frugal trial design, definitions, terminologies, looking at what was covered in those reading assignments a bit. And then a lot of this unit, we're going to be listening to a webinar that was recorded at the beginning of live seeding about participatory on-farm cultivar evaluation and the live seed project, and what was sort of learned from that. And then if you're actually joining this sort of in real time, you'd go on to this general discussion part. So let's get started learning more about the need for organic cultivar testing. So the need for cultivar testing in organic systems is elaborated quite a bit in Ray et al. And this is a snip taken from that deliverable, which has some of the text included. You can see the paragraph above that figure, and there's also a figure caption. And the figure is just supposed to sort of illustrate how conventional and organic farming systems are different, and how cultivars might perform differently in organic and conventional systems. So the cultivars in that particular schematic are represented by the sort of green dots. And what you can see on the left is the conventional farming system. The blue line is just representing environment, and that's just showing in conventional and organic systems there's lots of variations in environments, right? We know that soils are different, climate is different, pest pressures might be different, so you have variable environments in both cases. But in conventional systems, we use chemical inputs or synthetic inputs like fertilizers, pesticides, to moderate that variability and basically sort of mitigate it and bring performance up to a fairly uniform level by adding, more fertilizer, for instance, in cases where soils are less fertile, or a specific, fungicide in a place where there's more pressure from a specific disease. Low input systems and organic systems don't have those opportunities, so there tends to be more variability in outputs. But what's particularly important is about varietal choice, and how the varieties that rank highest in a conventional system may not be the same ones that rank highest in an organic system. And you can sort of see here, they're just illustrating, for instance, here's a variety actually higher in an organic system than under conventional production, quite a few lower. And so one of the justifications for doing decentralized distributed variety testing in organic systems is that those varieties that perform best in conventional may not be the same ones that perform best in organic, because of these very different management conditions and how we sort of deal with environmental variability. So that's illustrated there. This is also summarized here in this set of bullet points, just re-emphasizing the diversity of organic farms, and also that they're often smaller, more diversified in terms of crop rotations and environments and varieties that they grow, and the need for seeds that are adapted to the local management practices and environment. So this requires what we call post-registration cultivar evaluation trials. So testing of cultivars after they've been developed by breeders and they're registered already, they're on the market, but we need to know more about which ones are the ones most suited to the organic systems. And here also just again showing, this is also described in the webinar, but I thought I'd illustrate it here as well. The more conventional approach to cultivar evaluation, sort of starting from research at the top, generates lots of diversity, which are then selected under certain conditions and selected for the best performance under those conditions, which tend to be which are conventional management usually, although we do have organic breeders as well. Registered by the authorities, evaluated more by this agricultural knowledge information system used on farms, etc. And just pointing out that this sort of bit in the middle here can be quite heavy on logistics, time, infrastructure, and cost to ultimately come up with the cultivars the most appropriate for farmers to use in a given environment. And here's yet another illustration too, just to, again, showing, and this comes from the webinar, so it'll be explained there as well, possibly better than I'm explaining it now, but just showing you cultivar information obtained through trials in conventional settings. But we know that cultivar performance in organic isn't always the same as conventional. And what we're getting at there is really about ranking of cultivars, the cultivars that perform the best under conventional management, when some of those pressures, such as weeds, for instance, and sometimes fertilizer, are not present. So they're provided they're evaluated in conditions where there is weed control used and nutrients supplied. You may not get the same cultivar performing best as you do in an organic system. So the one approach would be to reproduce this sort of conventional cultivar trials in organic context. So just go out and perform many mini plot trials on a limited number of farms, but under organic conditions. But the sort of argument against that is that organic farming is highly diversified. So you'd need lots of environments. And of course, we have lots of species that need trialing as well, right? So it would become quite expensive and resource intensive, really, to do that. So organic cultivar testing might need more trials than in conventional systems. So that's why we need new cultivar testing models for organic farming, and that's why we've come to this idea of participatory and decentralized or distributed trials using these principles of frugal innovation. So that's again explained a little more in the webinar. And this is again showing you some illustrations of the alternative model for organic, decentralized, distributed trials, where you go from this pipeline on the left to more of an ecosystem approach, where you've got breeders, researchers, farmers, the value chain, the agricultural knowledge information system, etc., all interacting in sort of a network, as that large image on the right. And then just in the middle there, some different types of networks, just showing you a centralized network on the left. This is a bit more theory on it, really, from a paper quite some time ago, 1968. Centralized network, decentralized network, and then a truly distributed network, which is sort of like the ideal that we like to think we're aiming for, which is really sort of gives the farmers sort of the more power and control and autonomy. So frugal innovation, that's a principle that we want to talk about a little more when we get to sort of the design of these distributive, decentralized cultivar evaluation networks. So frugal innovation comes from business and technology world. It isn't necessarily applicable only to agricultural systems. It's about creating high quality solutions with minimal resources, focusing on cost efficiency, accessibility, simplicity, and scalability. So in the Live Seed project, so as I said, the predecessor to live seeding, there's a real focus on these 3 steps. So defining objectives, identifying constraints, and then applying appropriate methodology. Sorry, my picture's obscuring that last box, but that's applying appropriate methodology. And these are sort of 10 principles of frugal innovation that sort of underline these three steps. The solution is in the problem. Simplify what's complex, so keep it simple. Think about solutions that are sustainable and accessible. So these are all the principles that should be kept in mind when designing a on-farm cultivar evaluation network. So of course, not reinventing the wheel. So think about resources already to hand and how those can be used within your cultivar evaluation network. Use new technologies as a lever. So we're going to talk more in some of the later modules about some of the technologies like apps, for instance, for data collection. So don't be sort of afraid of those. We're not talking about not embracing new technologies. We're talking about using them to really kind of democratize this whole process of cultivar evaluation. Co-creation, so working directly with the end users along the whole value chain. Constraints there, and that's one that you can see was pulled out by the Live Seed team as one of the sort of key steps in the frugal innovation process. They advocated in Live Seed. And a few others there you can see. So sort of 10 principles to keep in mind, especially when we're looking at case studies later. Interesting, just to put this one in, this is a new representation. So that previous slide, I had it as a sort of a linear process, starting with defining objectives, identifying constraints, and applying an appropriate methodology. More recently in the live seeding project, we've realized that it's really an iterative process where you sort of may go back again and again during the whole process, the whole period of on-farm cultivar evaluation and the evolution of those networks to redefine objectives and identify possibly new constraints and adjust methodologies. So that's why I've just represented these three sort of steps now in a bit of a cycle. So we've come to realize it's not linear, finite, it's more of an ongoing iterative process. So yeah, now we're going to go to the video part of this unit. So this is a chance for you to watch a longer video covering a lot of those topics I just touched on in this intro section. And there are these questions that I'd like you to download from the platform you're using to take part in this module, and sort of follow along and try and fill in the answers to those questions, type them or write them in a notebook, and then at the end we'll review those. Now this is really where the learning becomes interactive. And I'll just go to the next slide. So it's just, open the file with the directed questions, fill in the answers as you watch the video. And that link will be a hyperlink if you're using the PowerPoint version of this presentation. So yeah, if we're doing this in real time, we will then have a sort of live discussion about these succeeding questions. If you're doing this sort of independently, then you'll just have to watch the video and have a little bit of a read and think about those different questions. You can follow through the PowerPoint afterwards if you want, which has some prompts to help you think about those questions. And then we do have sort of I don't want to say an answer sheet because there's not right or wrong answers to a lot of those questions. They're discussion questions, but we do have an answer sheet that you can download after that gives you some more sort of points on those discussion questions. So now just go ahead and have a watch of that video and follow through with the questions. And then, as I said, if you're doing this independently, you can have a look at the PowerPoint and the discussion prompts after, and then rejoin for Unit 2 of this module.